Hi everyone, this is your instructor Kwame Shen. This is the second part of our lecture on microbial metabolism. And so we'll look at the different biochemical pathways that we have in prokaryotes and some eukaryotes as well. But before we do that, let's do a quick recap of metabolism. So metabolism is a sum total of all the chemical reactions that occur within living organisms and help them to maintain life, carry out the various processes that they need in order to thrive as living organisms. There are two basic types of reactions that come together and are described as metabolic processes. And those are catabolism and anabolism. So catabolism is a reference to all the metabolic processes that results in the breakdown of complex organic molecules into simpler ones that the body can use to build new molecules. Now, in the process, energy is produced as well. We call these processes hydrolysis reactions because water is used to break down bonds. So if you look at what we have here in terms of, so you have a polymer and water is being used to break down these bonds. So you have OHH being introduced where the bond used to be, and the same for this as well. And this is the process by which complex molecules are broken down in cells by the process of hydrolysis. More energy is produced than it is used in breaking down these bonds. And so reactions of such nature are described as hexagonic reactions. That energy that is produced is then used for anabolic processes. Anabolism is when complex molecules are built or created from very simple ones, and it uses the process of dehydration synthesis. Dehydration synthesis is when water is taken out of these molecules, the simpler molecules. Water is taken out in the process as they are joined together. So you have OHH taken out over here. You have the oxygen rearranging and becoming part of the bond as it forms a disaccharide over here. In such reactions, in anabolic reactions, more energy is expended than it is produced, and so they are described as endogonic reactions. Metabolic processes, it's like a chain reaction. One feeds the other. So a catabolic reaction is connected to an anabolic reaction. And what I mean by that is this. So you have simple molecules like glucose, amino acids, and these molecules are then joined together to form complex molecules. In the process, heat is released, energy is used. So ATP is then used, and then it becomes ADP plus phosphates. Now, once you have these complex molecules, it gets to a point where the cells will need the energy that is stored in these molecules, or would need the smaller subunits of these complex molecules to build new components of the cell. And for that to happen, the bonds are broken. And when bonds are broken, ATP is created, or energy is released, and we end up with simpler molecules. So that's how the reaction occurs. The reactions that occur in metabolic or anabolic processes are described as redox reactions or oxidation reduction reactions. And by oxidation reduction reaction, what we mean is that these are reactions that take place together. So we describe them as coupled reactions, but you need both of them together for it to work. So you have two reactants. One reactant is releasing energy, the other is accepting, one is releasing electron, the other is accepting electron. Now, and with the electron comes energy, by the way. But yes, just the release of electrons and the acceptance of electrons. The molecule that releases the electron is said to have been oxidized at the end of the reaction. And the molecule that accepts the electron is said to have been reduced at the end of the reaction. So we have an oxidation component and a reduction component of the reaction. Now, in all these, it comes down to how energy is created, is carried, and then is used or reused by the cell. The main energy currency of cells is ATP, adenosine triphosphate. That's the most common way that energy is carried within cells of living organisms, including microbes. There are other forms of energy carriers, like nicotinamide adenine dinucleotide, NAD. You have the reduced form of it, and then you have the form that has the phosphates as part of the molecule. In addition to the removal of electrons in a redox reaction, you also have the removal of hydrogen atoms. So the molecule that is releasing the electron, it also is also losing hydrogen atoms. And the molecule that is accepting the electron is also accepting hydrogen ions. So that's another component of the redox reaction as well. In practical terms, for microorganisms to break down glucose, a redox reaction has to happen. Now, what happens is that the glucose molecule has hydrogen atoms in it. So as it loses electron, it loses some of these hydrogen atoms as well. And when it loses electrons and the hydrogen atoms, it becomes oxidized. So the glucose is oxidized. The oxygen, which if the organism is an aerobic microorganism, would use oxygen to break down glucose. The oxygen will accept this electron from the glucose and will also accept the hydrogen atoms from the glucose. And so the oxygen then becomes reduced, become water. The glucose becomes carbon dioxide. But the most important component is that at the end of it, you have ATP being produced. Because molecules are being broken down, you have more energy produced than is used. How then is this energy stored in ATP? ATP is an adenine nucleotide with an extra phosphate attached to it. So you have a third phosphate. Typically, you just have two phosphates, and you have three of them in ATP. So we call it adenosine triphosphate. The third phosphate carries a high energy bond. So how that energy is stored is that the energy is stored in the third phosphate. The bond formed between the rest of the molecule and the third phosphate contains that energy. That energy is hydrolyzed, so water is added and it breaks the bond. And once the bond is broken, you have the energy being released. But at the cell level, within these cells, how is this ATP created? How is it synthesized? And the generation of ATP will take one of these three forms, either by substrate level phosphorylation, what we call SLP, but phosphorylation refers to the process of adding phosphate to a molecule. So for instance, you have phosphoenol pyruvate, PEP, which is a substrate for certain microorganisms. And this is the enzyme that's going to catalyze the reaction. What the enzyme does is that it takes away this phosphate from the substrate and adds it to ADP to become ATP. And so energy is then created this way. And then you have pyruvate as the products. Okay. So this is an example of a substrate level phosphorylation. You have a substrate, you have an enzyme that acts on the substrates, and then you have ATP being produced. Then there's a second type of ATP generation, which involves the use of oxygen for aerobic organisms. For anaerobic organisms, you may have sulfur, phosphorus, or carbon being used, carbon dioxide being used as the final electron acceptor. Now, the second one is called oxidative phosphorylation. It occurs in electron transport chain. We'll talk about it briefly. But for that to happen, you need a final electron acceptor. So that's the second way of synthesizing ATP. Then there's a third manner in which ATP is synthesized, and that is known as photophosphorylation. So light is used to convert chemical energy in the form of ATP, ADP to ATP. What happens is that you have, for instance, photosynthetic microorganisms that will trap the sun's rays. It will lead to the excitement or the removal of electrons. And so when these electrons are removed, then they move through the different molecules that exist within the chloroplast. You end up having a redox reaction, and so the electrons are accepted, and in the process you have ATP produced. So ADP plus phosphates, you have ATP. So this happens in photosynthetic microorganisms. So the three ways by which you can create ATP is you have substrate level phosphorylation, where you have a substrate and an enzyme. You have oxidative phosphorylation, which happens in electron transport chain, where you need a final electron acceptor. Then you have photophosphorylation, which is part of the process of photosynthesis, where light is used in the synthesis of ATP. For the metabolic pathways, the breakdown of complex microorganisms, we should note that to produce energy doesn't happen at a go or in just an instant. If it happens in just one reaction, what will happen is that you have too much energy released that will result in the destruction of that microorganism or the cells of the body in terms of eukaryotes. However, what happens is that it's rather a stepwise process. So you have a material that's a starting material A, there is a conversion of it to B, and there is a conversion to C and to D. So it's a stepwise process and not just one reaction that takes place at a go. It's protective in this nature. So that's a very important point that you should know. Carbohydrate is one of the commonest source of energy for many living organisms. So we'll look at glucose metabolism. And so in this case, we're talking about catabolism, because the breaking down of glucose. It involves these processes. So we'll talk about this process. We have the process of glycolysis. And after glycolysis, you have pyruvate or pyruvic acid that is synthesized. Pyruvic acid will then go into either the Krebs cycle and the electron transport chain, or it will go into fermentation based on what's available and what the microorganism is capable of doing. Glycolysis is the first step of glucose metabolism in most living organisms. And essentially, it is the breaking down of a 6-carbon glucose molecule into 2 3-carbon pyruvic acid molecules. So this 3-carbon molecule is either called pyruvic acid or pyruvate, and they are used interchangeably. Now in the process, you have 2 ATP that are produced, net energy, and then you have 2 NADH that is produced. NADH is reduced, the hydrogen makes it reduced, so it reduces nicotinamide adenine dinucleotide. In a more broader sense, Glycolysis involves these steps. So you have a 6-carbon molecule that is phosphorylated, and in the process of phosphorylation, ATP is used to form fructose diphosphates. This 6-carbon molecule is then broken down into 2 3-carbon molecules called glyceraldehyde 3-phosphates. Now, so you have 2 glyceraldehyde 3-phosphates, and each of them would then undergo various transformations so that you end up with pyruvate. So you end up with 2 pyruvic acid molecules. Now, in the process, you have phosphates that are added. There is rearrangement that takes place. It results in the synthesis of NADH and 2 ATP for each molecule of glyceraldehyde 3-phosphates. So that for one glucose molecule, you have one NADH produced by one glyceraldehyde 3-phosphates. You have another, so this times 2. You have 2 ATP times 2. So you end up with 4 ATP that are formed. But note that you already used 2 ATP at the beginning. And so if we take away the 2 that we use at the beginning, you end up with a net of 2 ATP. So glycolysis produces a net of 2 ATP. Glycolysis produces 2 pyruvic acid or pyruvate. It produces water as well. And then it produces 2 NADH, 1 from each half. So let's do some accounting of what's going on in glycolysis. What's the purpose of glycolysis? The whole purpose is to break down glucose into pyruvic acid and to release energy, to produce energy. Where does it happen? It happens in cytoplasm of all cells, both eukaryotic and prokaryotic cells. What are the reactants in this process? So you have glucose, you have ATP, because ATP is used at the beginning, and then you have NAD+, which is then converted into NADH. So what are the products? You have pyruvic acid, you have 2 of them. You have ATP, you have 4 of them. You have NADH, you have 2 of it. But the net energy gain is 2 ATP and 2 NADH. 2 ATP because you already used 2 here. You subtract the 2 from the 4 that is formed, you end up with 2 ATP. In addition to glycolysis, some microorganisms use other pathways to break down glucose. And the 2 commonest ones that we'll talk about are the pentose phosphate pathway and the Entner-Dodorov pathway. The pentose phosphate pathway is also known as the hexose monophosphate shunt, and it operates simultaneously. So as glycolysis is going on, the pentose phosphate pathway is also going on. So that's one feature of it. It results in a breakdown of pentoses. A pentose is a 5-carbon sugar. So it breaks down of pentoses and glucose. It will produce intermediate compounds that are then used in the synthesis of nucleic acids and glucose, etc. Another point is that there is a net gain of 1 ATP for each glucose molecule that is oxidized. Compared to glycolysis, there is a net gain of 2 ATP. So that's another difference that you see there. You have Escherichia coli or E. coli, Enterococcus faecalis, and Bacillus subtilis as some of the microorganisms that use the pentose phosphate pathway. Then you have the Entner-Dodorov pathway. The difference between the Entner-Dodorov pathway and the pentose phosphate pathway is that for the Entner-Dodorov pathway, it can operate without glycolysis. Okay. It can operate by itself, independent of glycolysis or the pentose phosphate pathway. It produces 1 NADPH, 1 NADH, and 1 ATP. Note that it also produces 1 ATP for each molecule of glucose that is oxidized. This is generally common in gram-negative bacteria such as Pseudomonas and Rhizobium. And we don't usually find this process taking place in gram-positive bacteria. So it's one of the ways that you can use to separate the two. Sometimes in clinical settings, they are used to identify Pseudomonas. So when glycolysis has taken place and you have pyruvic acid or pyruvate present in the cells, pyruvic acid is toxic to the cells. The cell has to find a way to transform this molecule into a less toxic form. And it has two options available. If there is no oxygen present or no electron acceptor present, it goes into fermentation. So it can either take the pathway of fermentation or it can enter respiration. Okay. So you have the respiration could either be aerobic respiration or anaerobic respiration. But those are the options that it has. Now, if the pathway is cellular respiration, then the pyruvic acid will be converted into a less toxic form called acetyl coenzyme A. It will then enter the Krebs cycle, and then it will enter what we call the electron transport chain. This is what the Krebs cycle entails. And so you have the pyruvic acid or pyruvate from glycolysis, which is a 3-carbon molecule, would then be transformed into a 2-carbon molecule called acetyl-CoA. Acetyl-CoA would then enter the Krebs cycle and then it to be transformed. In the process, you have carbon dioxide being produced at the first step. You have other carbon dioxide molecules produced also here, 2 of them. You have NADH, 3 of them. You have FADH2, and then you have ATP also synthesized. So I'm trying to go step by step in terms of the Krebs cycle. The first step, which technically is not part of the Krebs cycle, is called a preparatory step. It's the first step in the process. So you will have pyruvates, and pyruvates will then be converted into acetyl-CoA. You would have CO2, CO2, so the carbon and 2 oxygen atoms would then be taken away. That's CO2. You have NAD plus that will be converted into NADH, and you have coenzyme A that will then come and bind to the rest of the molecule that is left. So you end up with acetyl-CoA, which is a 2-carbon compound. So you have your first carbon dioxide produced here, the preparatory stage. And then in Krebs cycle itself, when acetyl-CoA, which is a 2-carbon compound, enters the Krebs cycle, it combines with oxaloacetate. Oxaloacetate is a 4-carbon compound. And so a 4-carbon compound combines with a 2-carbon compound, you end up with a 6-carbon compound called citrates. Citrate then undergoes some transformations and becomes isocitrate. So you have isocitrate formed here. Isocitrate then loses 1 carbon as carbon dioxide, and in the process you have NADH produced, and then it becomes alpha ketoglutarate. Alpha ketoglutarate also loses 1 carbon atom as carbon dioxide, and in the process you have NADH also produced. And so alpha ketoglutarate then becomes succinyl-CoA. Succinyl-CoA would also undergo transformation and become succinate. In the process you have ATP produced. Succinate would then undergo transformation and then become fumarates. Fumarates, in the process you have FADH2 produced. FADH2 is reduced flavin adenine dinucleotide. It will also undergo transformation and becomes malate. All this time, it is still a 4-carbon compound. It is just rearrangements that is going on. In the process, you also have NADH produced again. And the final product is a 4-carbon compound called oxaloacetate, which will combine again with acetyl-CoA, and the cycle continues. But keep your eye on this. You have 2 carbon dioxide molecules that are produced. You have 1, 2, 3 NADH molecules that are produced. You have 1 FADH 2 molecule that is produced. However, this is for just 1 pyruvic acid molecule. For every glucose, you have 2 pyruvic acid molecules that are produced. So you just have to double all these in order to really account for the molecules that are produced per oxidation or per metabolism of just 1 glucose molecule. And that's what I mean. Then the preparatory step, you have pyruvic acid becoming acetyl-CoA. We are accounting for the products in terms of the byproducts and the energy carriers. So the byproducts, you have CO2 that is produced, 1, and you have NADH, which is an energy carrier, which is also synthesized. And then it enters the Krebs cycle. So you would have CO2, another CO2, that's 2 of them. For 2 cycles, you have 4 CO2. You have 1 ATP, since you have 2 pyruvic acid each of them going into these processes. You have 2 ATP. You have FADH2, you have 1 of them times 2, so you have 2 of them. NADH, you have 3 of them, 1, 2, 3, times 2, you have 6 of them. So for Krebs cycle, if we take away the preparatory stage, you have 1 pyruvate, you have 3 NAD plus, you have 1 FAD plus. And so they are transformed into NADH, you have 3 of them, FADH2, 1, CO2, you have 2 of them, you have ATP, 1 ATP. So these are the things you should remember. For glucose is converted into pyruvate or pyruvic acid. In the process, you have 2 NADH that is produced, you have 2 ATP that is produced. Each of them will go into its separate cycle, and in each cycle, a pyruvic acid will be converted into a less toxic form, acetyl-CoA. In the process, you have carbon dioxide and NADH, and then also the acetyl-CoA will then enter the Krebs cycle, and once it enters the Krebs cycle, you have production of 3 NADH, 1 FADH2, and 2 carbon dioxide. Okay. So if we are accounting for all the 2 processes together for each glucose molecule, you have 6 NADH, 2 FADH2, 4 carbon dioxide, and 2 ATP. However, if we add a preparatory step to it, add a preparatory step to the whole process, what you end up with is that you have 2 ATP, which is produced in the Krebs cycle proper. You have, in terms of NADH, you have 6 produced in the Krebs cycle itself. You have 2 produced in the preparatory stage. You have 2 FADH also produced in the Krebs cycle. Note that the Krebs cycle is also known as the citric acid cycle or the tricarboxylic acid cycle. Then you have the electron transport chain. The molecules that are produced here in Krebs cycle, you have NADH, you have FADH2. These are the most important products in Krebs cycle because they contain the most energy that was originally contained in the glucose molecule, those chemical bonds in the glucose. So the most important molecules in terms of the products of Krebs cycle are NADH and FADH2. And that's an important point that you should know. So that takes us to the electron transport chain. The electron transport chain, the purpose is to harness the energies in NADH and FADH2. That's the whole point of it. And where does it take place? In eukaryotes, it takes place in the mitochondria of the cells, and prokaryotes. Note, prokaryotes do not have mitochondria. So for prokaryotes, it takes place in the plasma membrane. It starts in the cytoplasm or the cytosol, but it actually ends in the plasma membrane. Now, the condition for an electron transport chain to occur is that there should be a collector of electrons. For aerobic organisms, the collector of electrons is oxygen. For anaerobic organisms, microorganisms, they will have other sources of electron collectors or electron acceptors, like sulfur and carbon dioxide and methane and the likes. Well, what are the reactants then? The reactants are NADH and FADH2, and the products are NAD plus and FAD plus 2. So the energy is stored in electrons. The electrons that we find in NADH and FADH2. Now, the cell has to find a way to remove this energy. And since the energy is stored in electrons, at the end of the process, the electrons has to be captured. It has to be accepted by another molecule. So this brings us to the electron transport chain. The electron transport chain is a system made up of a series of carrier molecules, and some of these carrier molecules are hydrogen pumps. So those are depicted in blue over here. These are not hydrogen pumps, but they are carrier molecules, and so they're just carrier molecules but non-protein in nature, and they are also part of the electron transport chain. In addition to that, there is an enzyme that forms part of the electron transport chain known as ATP synthase. The ASE indicates that it is an enzyme. It is, however, enclosed in a membrane as shown here. Now, the electron transport chain in prokaryotes occurs in the plasma membrane. So, plasma membrane. In eukaryotes, it occurs in the mitochondria. And the mitochondria, from previous knowledge, we know has an outer membrane and an inner membrane as well. So we have that's the outer membrane, OM. I'm just going to use OM just to shorten it. Then we have the intermembrane space, IS, and then you have the inner membrane, IM, and in here you have the matrix. It is in the matrix that Krebs cycle occurs. Okay. Now, it is this membrane here, the inner membrane, that has been enlarged over here in this larger diagram. So this would be the inner membrane, this would be the matrix, and this will be the intermembrane space. The outer membrane is not depicted here. So you have charged molecules from the Krebs cycle, NADH and FADH2, with very high energy electrons in them, and these have to be harnessed in order to synthesize ATP. Now, for that to happen, these molecules are oxidized. So they are oxidized by the removal of the electrons. The electrons are removed because molecules generally want to have a more stable state than they are. They always are trying to have a more stable state. So these high energy molecules, NADH and FADH2, are oxidized. And when they are oxidized, electrons are released and hydrogen atoms or hydrogen ions are also released, or protons are released. Remember, we already have hydrogen pumps over here. And these hydrogen pumps would then pump the hydrogen that are released from the NADH and FADH2 into the intermembrane space. Now, it gets to a point where you have an imbalance of positive charges. You have more positive charges in the intermembrane space than in the matrix. And then you also have more hydrogen on this side in the intermembrane space than in the matrix. And this is an example of what we call an electrochemical gradient. Electrochemical gradient. Okay. The electrochemical gradient that is set up means that there are at least one condition here that has to be reverted, in that you have an imbalance of positive charges and hydrogen molecules as well, hydrogen atoms, sorry. Now, this is an example of potential energy, in that these are stationary in the intermembrane space, but once you get them moving, you can generate energy, and that's exactly what happens. However, these protons can only go back into the matrix, can re-enter the matrix through the ATP synthase complex. Now, the ATP synthase is an enzyme, and so whilst these protons are going through the ATP synthase complex, energy is generated because they're moving now, and that's kinetic energy. This kinetic energy then serves as activation energy that catalyzes this reaction, the addition of ADP to pyrophosphates. And so ADP is added to phosphate, or phosphate is added to ADP, and you end up with ATP. So adenosine diphosphates end up becoming adenosine triphosphates. This is the energy currency of the cells, and this is the end goal of the entire process. So this process is an example of chemiosmosis. Chemiosmosis is basically the synthesis of ATP from an electrochemical gradient. And so ATP synthesis using electron transport chain, as I just described, is an example of chemiosmosis. Now, we don't have to lose sight of the fact that the electrons are still in the system, so it has to end up somewhere. Now, these electrons will go through the carrier molecules, electron carrier molecules. So we'll go through the pumps and the other molecules that are non-protein in nature. And as they're going through it, it's a cascading reaction. They move from a very high energy level to a lower one, to the next lower one. Eventually, when they exit the membrane, they have to be accepted. They have to end up somewhere. And in aerobic organisms, for aerobic respiration, they end up being accepted by oxygen. So we say oxygen is a final electron acceptor. And oxygen accepts these molecules. So the oxygen that we breathe in ends up accepting these electrons. It splits it up into two atoms of oxygen, and one of them would then bind with the hydrogen that is present in the matrix, to form water, which is one of the end products of respiration. Now, for anaerobic organisms, these electrons are accepted by other molecules other than oxygen, like sulfur, carbon dioxide, methane, and so on and so forth. So this is our participation question for the week. What conditions are necessary for the process of chemiosmosis in electron transport chain? And your response should include at least one importance of chemiosmosis in cellular respiration. And it's also important that you state your response in your own words. And so even if you do some research around, read around and all that, write these in your own words. And that's how I can really tell that you understand what's going on. So for each NADH hydrogen atom that is recycled, you have 3 ATP that is produced. For each FADH2 that is processed, you have 2 ATP produced. In aerobic respiration, the terminal electron acceptor is oxygen. Okay, and in the process, you have, in prokaryotes, you have 38 ATP that is produced. That's a lot of energy for one molecule of glucose. However, in anaerobic respiration, these microorganisms will use sulfur or nitrates or carbonates as the terminal acceptor, but it's not as efficient in terms of the amount of energy that is produced. So by review, you have glucose. It's converted to pyruvate, 2 for every glucose molecule. So that's a process of glycolysis. You have ATP and NADH produced. And then you have the pyruvate that will then be processed into a less toxic form. In the process, you have NADH and carbon dioxide released. So you have acetyl-CoA. It will then go into Krebs cycle. In the Krebs cycle, you also have NADH, FADH2, and carbon dioxide that are produced. And then you have these molecules then entering the electron transport chain and undergoing the process of chemiosmosis. And when the gradient is established, and the protons have to go through the ATP synthetase, you end up having ADP being added to phosphates, and it's catalyzed to form ATP. In prokaryotes, you have 38 ATP molecules for one glucose molecule that undergoes aerobic respiration. In eukaryotes, you have 36 ATP molecules that are produced as a process of the breakdown of glucose. Fermentation. So if oxygen is available, aerobic respiration will take place. If oxygen is absent, fermentation will take place. Fermentation may take place in mussels or it may take place in yeast. In mussels, you have a less toxic form of pyruvic acid formed, known as lactic acid. In yeast, where you have alcohol fermentation, you have carbon dioxide and ethanol being produced. And so the whole point of fermentation is to process pyruvic acid into a less toxic form. Okay. And since oxygen is not there as the final electron acceptor, pyruvic acid acts as a final electron acceptor. It doesn't require oxygen. It recycles NADH to NAD+. It doesn't require Krebs cycle or the electron transport chain. Remember, it only takes that route in cellular respiration where oxygen is present, or there is a final electron acceptor. It produces a small amount of ATP, and so you will notice that the ATP that is produced in fermentation is just the ATP that is produced during glycolysis. So no ATP is technically produced during the process of fermentation. And most of the carbons stay in the same form as the in the organic compound at the beginning. So if we look at the types that we have lactic acid fermentation, which occurs mostly in cells. It results in the synthesis of NADH, two of them. And then we also have alcohol fermentation. And for alcohol fermentation, you have the production of carbon dioxide. So you have a 2-carbon molecule. And then there is rearrangement, and NADH is produced, 2 of them. But in the end products, you have alcohol as the end products. So let's look at accounting for the energy in terms of fermentation. There's a breakdown of the toxic pyruvic acid. Fermentation occurs in cytoplasm of cells. Okay. The reactants, your pyruvate and NADH. For the products, you have a less toxic compound. It could be lactic acid or it could be alcohol. And then you have NAD+. You have one of it that is produced. But then you may have CO2 depending on what kind of fermentation it is. In alcohol fermentation, there is CO2 that is produced. In lactic acid fermentation, there is no CO2 produced. We have a lot of microorganisms that ferment different products. And so we end up with all these products that we use in our daily lives as a result of fermentation by microorganisms. So these are some examples where you have Saccharomyces, which is used in the production of beer, wine, and the likes. And the end products there is carbon dioxide and ethanol. And then you have lactobacillus or streptococcus producing lactic acid, which we enjoy as yogurts and soy sauce and cheese. So breaking down it again, when cellular respiration takes place, there is a lot of more energy used. Oxygen may be the electron acceptor. Other molecules in anaerobic respiration would need different molecules as the electron acceptor. And it is thought of to be one of the processes that evolved after fermentation that is more sophisticated than fermentation. And then there's fermentation, very little ATP is produced, only 2. It doesn't need oxygen, and it's thought of to have evolved first. And pyruvate is seen as the electron acceptor in that process. So if we look at the overview of the whole process in terms of the different pathways. When oxygen is present, you have 36 to 38, depending on which cells you're looking at, produced. Anaerobic respiration, you have 2 to 36. You have different electron acceptors, so electron acceptors other than oxygen. You have sulfates, nitrates, carbonates that are used. And then there is fermentation, that releases only 2 ATP. Okay. There are other sources of energy aside glucose, so you may have fats or proteins, and they will enter different points in the metabolic pathway. So lipids will be converted into glycerol and fatty acids. If it's glycerol, it could enter the glycolysis stage and continue there. Your fatty acids may enter the intermediate stage, and for proteins, broken down to amino acids may enter the glycolysis, the intermediate stage, or may enter the Krebs cycle stage. So that brings us to the end of part 2 of our lecture on microbial metabolism.